So let's move on to the next talk for the session. This is by Lucie Delemotte. She is heading the SciLife lab at KTH Royal Institute of Technology at Stockholm. And the title of her talk is Conformational Landscapes of Membrane Proteins. So I
thank
think,
you so
Christine,
much.
you will have, yeah, there you go.
And thank you very much for organizing this event. I concur with everyone. It would have been very nice to be able to do this in person, but I'm happy that we get to talk together around this really interesting topic of rare events, of course. So I wanted to discuss membrane proteins, and of course the topics are sampling rare events, but I'll be focusing on a slightly different type of system. And so my lab is very interested in membrane proteins. Historically, we worked quite a lot with ion channels, which specifically voltage-gated ion channel that enables cells to carry electrical signals. And more recently, we moved into other types of proteins. And this talk will be especially focused on G protein coupled receptors, but we also have interest in other classes of proteins like sugar transporters. And in terms of what we're interested in specifically, the questions that we're looking at are really quite related to biological applications, pharmacology, and so on. One main area of application that we're working with is regulation. So can we understand how ligands, lipids, drugs, post-translational modification, auxiliary proteins, and so on, how do they modify the function of proteins, and going into the areas of disease and drug discovery. But before we do all this, we actually have a first step to overcome, and that is understanding the biological function in absence of any type of modulation. And to be able to do this, as you can tell from the little pictograms that I had, one important thing is to understand the conformational cycling. And that means how do we go from specific functional state to other specific functional state. And of course, this is really pretty difficult to achieve with molecular dynamic simulations, mainly because of the timescale problem. And that has been introduced quite nicely in many talks before this one. So here I'm showing you an example of one type of protein that we work with. That's the voltage-gated ion channel. And starting...
Sorry, excuse me, Lucie. Are you changing the slide? Because from our side, the slide is not changing.
I see. It says sharing is paused.
Oh, sorry. I'll ask. Can I ask Nishant to?
Yeah, I think you can unshare and share again.
Right.
I think what happened is that somebody entered the waiting room and somehow that stops my screen sharing for some reason.
Okay, so, can you try going back one slide and moving back
now?
one? Yeah.
Yes, yeah.
So that you can see the movie playing also?
No. Can you go back one slide and come back to the slide so that we can see if it's the slide transition?
What do you see now?
No, I mean, we are seeing the, what is it?
Sorry, let me try again.
Yeah.
Hopefully no one enters the waiting room. How about now?
Yes, we are seeing the voltage-gated channel. Yeah.
Okay, thank you so much. I'm sorry. So what you see exactly on here is a voltage-gated ion channel being activated. So we start here from an activated conformation that was resolved by cryo-electron microscopy. And then over time, we can see the response of the system to an activating voltage. And this is, of course, a really nice case where we're able to see a very big conformational change. But unfortunately, this is rather the exception. And in fact, to be able to observe this, we first had to apply a very big voltage, transmembrane voltage of around 0.5 volt. And also, we were running on Anton, the supercomputer that is especially designed for ND simulations by D. Shaw. And most of the time, we, of course, don't have access to such resources. So I see I'm having the problem again with sharing being paused.
Maybe we can go back to just displaying your screen, I think.
So what is in the chat?
Oh, it says
I was sharing the appli. Should I share the entire screen? Is that what you're telling me?
I mean,
yeah, Lucie, I think you should share the entire screen.
Yeah.
Instead of the slides.
Okay, so here now it's fine.
Yes, it's better.
Yes,
okay.
now it's working. Now it's working.
Fine.
Yeah.
Okay. So this is all motivation to say that most of the time, if we're interested in such complicated conformational change, and we don't have access to Anton simulations, then we need enhanced sampling protocols to sample the full relevant conformational ensemble. And I believe nobody needs really to be convinced of that in this audience. But so a few years ago, I got into this topic of trying to find the appropriate enhanced sampling method for the systems I was interested in. As I like to share my problems and wisdom, I was discussing with many people what enhanced sampling method I should use. And as I was doing this, it was quite interesting. I realized that many people have really different opinions. And what was happening most of the time is that I was discussing with people who had developed enhanced sampling methods, and they tended to tell me that I should be using, in fact, their method, which was, a better method than the next one. So going through this, I started thinking maybe it would be an interesting endeavor to try to see what different enhanced sampling methods exist, and could we build an exhaustive list, and are there really actually hundreds of different methods like it appeared to me when I was a postdoc, or was that sort of an artifact and many people were just rediscovering methods that were already published and kind of calling them differently, rederiving them using different notation and so on and so forth. And so that ended up turning into a research project that I've been doing together initially with Jérôme Ménard, who's in Paris. What we tried to do here initially was to basically itemize enhanced sampling methods and try to sort them. So this was a brainstorming kind of map that we came up with when he visited Stockholm in 2019. And basically what we did is that we listed all of the enhanced sampling methods that we had heard about or read about on these different post-its, and then we tried to see what they had in common and what was different about them. So as you can see, this is a pretty complicated map, but we pushed further with this project and tried to come up with a slightly better way of describing the landscape. And so the way we thought we were able to then categorize these different methods is to see what are the crucial branching points between that differentiate methods from one another. So in this decision tree that we ended up coming up with, we can see basically different so-called branching points or dichotomies that you answer each question basically with yes or no, and this takes you into a different type of method. So the leaves of the tree here in the boxes at the bottom represent different sort of classes of enhanced sampling methods, whereas those diamonds basically represent the dichotomies. And the thing is that there is no best way to sort of organize those questions. You could arrange them in many ways, and you would end up with a different tree every time. But we try to organize them according to what is the sort of fundamental differences. And maybe the most fundamental difference we thought was important is whether the original configurational distribution is preserved. And if so, we end up in methods that either use transitions to other ensembles, and those would be expanded ensemble replica exchange, or if they don't, those would be adiabatic methods. If we don't preserve the configurational distribution, then we can take the system out of equilibrium, and there we would find driven methods like steered MD. Or if we don't take the system out of equilibrium, we fall in the area of importance sampling. And then importance sampling is sort of divided into two main branches. Either we specify the starting coordinates and localize them either with localization methods like umbrella sampling or we just generate samples from different starting points, a little bit like Christina was describing, and that would be adaptive seeding, adaptive sampling. If we don't specify the coordinates, then we end up with these bias simulations where we can use a collective variable or not. And then the CV-based methods are maybe the ones that a lot of people are aware of, like metadynamics or ABF, whereas we also have a non-adaptive bias simulations that don't use a collective variable. And so we think this way of describing the method, and the paper should be posted on arXiv within a few days, is really quite interesting to make sure that we don't waste our efforts by using methods that are either not appropriate or using methods that are really similar in spirit. And so if one doesn't work, then the similar one is not likely to work. I should also mention when we were doing this, like I said, so the organization of the questions is a little bit arbitrary. And in fact, for example, the question of whether a collective variable is used also applies to other boxes. So for example, a diabatic methods usually would use a collective variable. And so if we had organized the boxes differently, then we would end up with that question also on that branch. So in the end, of course, this is described in the paper. There are sort of overlaps between the branches. And I should say, one thing we tried to do in this review is to redefine all of these formalisms using the same notation, because one thing that makes it difficult to compare the methods is that the formalisms and the notation is different from one paper to the other, making it difficult to compare. So anyways, with this, I felt a little bit more able to decide what would be a good enhanced sampling scheme. And I will now describe one specific effort we've been doing in the lab to study conformational sampling of GPCR. And so G-protein coupled receptors are membrane proteins that bind ligands to the extracellular side. And that triggers a conformational change in the receptor, which is a 7 transmembrane helices protein, which makes it possible for the G-protein to bind the GPCR to bind G-protein or other intracellular binding partners. And that triggers, of course, important reaction cascades in the cell. And this looks really fairly simple. And actually, this is a movie of the type of conformational change happening if you look at the receptor from the bottom here, where G-protein fragment comes and binds to the intracellular binding pocket as activation occurs. And this looks fairly simple, but in fact, in reality, the picture is really quite a bit more complicated, because instead of having only one resting state and one activated state, the conformational landscape is much more rugged, involves several resting states and several activated states. And so GPCRs are, of course, very important pharmacological targets, and they're also a huge family, which is represented on this phylogenetic tree. But fortunately, it's also a very well-studied protein family with many structures that have been resolved. And we focus here on a receptor that can be seen as a prototype, because it was one of the ones that was the most studied, and that's the beta-2 adrenergic receptor. And we have structures of it in several states, including activated and resting state. The resting state here is represented in white, and the activated state is represented in orange. So that puts us in a good place to try and figure out what the conformational ensemble linking those two states are. And just to introduce you to some of the nomenclature that I'm going to be using, the ligand binding site is located here at the top of the receptor. And these different labels here represent so-called microswitches, which are microscopic degrees of freedom that change upon activation. So the one at the top here, the TM5 bulge, is directly in contact with the ligand and contracts upon ligand binding. And then as we go down the receptor, we find a connector region, aspartate 79 region, the NPXXY motif, tyrosine YY motif, and the TM6 motif. And all of those basically sort of act in concert to propagate the ligand binding signal to the intracellular binding site. And I'll come back to how these microswitches come together to form this allosteric pathway. So if I go back to this question of what would be a good enhanced sampling technique, then we have to ask ourselves what we know about the problem, because depending on what we know about the problem, then we might have access to different types of enhanced sampling schemes. And so I should say, by design, we are in a really good state here because we have end states that were resolved experimentally of the active and inactive structures. And in addition to this, we also have a very good guess of an initial pathway, and that's because there were Anton simulations of the inactivation of this receptor that were published about 10 years ago by Ron Dror and colleagues. Now, on the negative side, what we know by scrutinizing this pathway is that the path that links the two states of the receptor appears really highly nonlinear. And this means likely that if we want to use collective variable-based methods, we need a very highly multidimensional CV space. And that means that there are many CV-based methods that we wouldn't be able to use. In addition to this, I should say something that I've been particularly concerned with is also computational efficiency and code availability. Code availability is for transparency reasons and being able to communicate our results in general with the idea of participating to open science. And computational efficiency is both for sort of ethical reasons, but also because we would like to be able to run these calculations many times in order to be able to study what I was mentioning early on, which is modulation by different ligands, lipids, and so on and so forth. So in that respect, we're quite interested in trying to find some methods that are pretty computationally efficient. And with that in mind, if we go through this tree, the method that we settled on belongs to this box it's one specific method where we localize the simulations by seeding the simulation strategically. And more specifically, we settled on trying to use the string of swarms method in high dimensional collective variable space. And the idea of the string method is that you start from an initial guess of a pathway that links two states. You basically steer the system along this path, and you then seed short simulations from all the points that are placed along this path. By looking at the average drift of these simulations, we can find the direction of the slope on the landscape, and then we can iteratively find the minimum energy path that links the two experimental states. And like I said earlier, one reason for using this or for choosing this method is that we have a starting pathway, a pretty good initial guess of the pathway, because there were inactivation trajectories that were published in 2011. Finally, like I was hinting at, we want to use a pretty high dimensional collective variable space because the conformational change is really highly nonlinear. And we do this by using machine learning on the conformational ensemble. And let me just detail this a tiny bit more. So we, as others, have learned from past experience that to pick out that in order to design to pick collective variables, it's usually a good idea to use data analysis to understand the system a little bit better. Prior to this, we were always using sort of just human intuition to infer collective variables, but we have since learned that it's usually quite good to use the machine to help us. And so, like other people, we have developed our own set of tools that allows to extract collective variables. There's a set here of non-supervised methods, both linear in terms of PCA and also autoencoder, like Christina was just describing, or a random Boltzmann generator. And then we also have classifiers here on the right, and I'll be talking a bit more specifically about classifiers. So for example, here we have multi-layer perceptron that predicts what kind of class we have, as well as random forest. And so in terms of the classifiers, because this is what we use the most to determine CVs, basically what we do is that we take the ensemble that contains our active state, our resting state, and maybe an intermediate state that makes 3 classes. And then we learn which input features are important to make the classification decision. And those would then be our CVs. And we've played around with determining what are good input features. In our case, we find that using C alpha distances that are modified as inverse distances works very well. And that's because the conformational change that we're describing are mainly dominated by contacts breaking and forming. For other systems, it might be important to use dihedrals, for example, but in our cases, we always find that the C alpha distances work well. So then we train these machine learning models to recognize the conformational states. And we look at the input features that were important for the classification decision. And for the moment, we haven't completely found a good way to uniquely determine which, where to set the threshold pretty much. So we usually do this with an arbitrary threshold, and in the system, the beta-2 adrenergic receptor, that means that we end up with a set of CVs that contains 40 distances that were important to classify between active, intermediate, and inactive states. So now we have an initial path. We have the CVs that describe the landscape we're interested in, so we can run the string simulations with swarms of trajectories. And without really boring you with details, I can say that in total, we run quite a few iterations, and we end up with about 4 microseconds worth of data for each of our ensembles, which is still can be considered quite a lot, but is really quite reasonable to obtain fully converged conformational ensembles for this sort of systems. This is just an example showing you projected onto 4 of the 40 CVs that we're interested in. We start from an initial path that, like I said, was based on Anton trajectories. And over time, the iterations proceed and we converge to a final energy path. And you can see that over the last 100 iterations, there is some drift in CV space or some fluctuation in CV space, but no drift. And that means that we have converged to the sort of path linking the 2 states, and we're sampling a thin sort of tunnel around this path. So with this, then we can take our ensemble, and then we basically discretize the space in CV space, and then we calculate the transition matrix in these on this little grid from the swarms of trajectories. Let's say the last, for example, 100 iterations. And then we can get from this transition matrix the free energy landscape, either by taking the top eigenvector, or we could also build a Markov state model and calculate the free energy. And when we look at this path here projected along 2 top collective variables, we can see contrasting the receptor with no ligands and the receptor with an agonist, that the pathway has changed. So instead of being quite diagonal in the space, When the receptor is bound, we tend to adopt rather a L-shaped type of structure. And in addition, we can also see that the binding of an agonist favors the active state, which is qualitatively what we would expect. So of course, that's very nice to see. We also mark the landscape with some important switches. For example, we can say that the twisting of the connector region occurs very close to the inactive state, whereas the switching of the other microswitches occurs much more in a transition state on the way to the active state in the absence of the ligand. And that is, of course, very nice, but we wanted to go a little bit further and see if we could characterize even better the modulation of the receptor. So now that we have a protocol for which we need to gather a few microseconds of data, we can run these string simulations for a variety of ligands. So here we have 7 conditions, one without ligand, and then agonist and inverse agonist, which should stabilize the inactive state, and agonist, which should stabilize the active state. And then we run the string simulations and characterize the free energy landscape to see that indeed the agonist stabilize the active state, which is marked by this R star, compared to the antagonist and inverse agonist. So at least on the qualitative level, the protocol seems to work. And then we asked 2 things. First thing was, is this only qualitative or do we have quantitative agreement? I'll come to that in a second. Another thing is that we can look even further, and instead of looking at just the microswitches here, we can then learn what the conformational ensembles exactly look like. And one thing that's already apparent on these landscapes, especially the one at the bottom here, which is a projection along the microswitch at the top of the receptor and the microswitch at the bottom of the receptor, is that the active state basin is dependent on the ligand. So the active state that is stabilized by this full agonist, you can see here, is quite different from the active state that is stabilized by adrenaline, the endogenous agonist. But I'll come back to that in a second. When it comes to looking at the potential for this method to generate quantitative insights, what we did is that we plotted the expectation value of the microswitches against the experimental measure of activation, namely the maximal G protein mediated CM production. And as you can see here, it's really interesting to see that we have a very nice correlation between the two, which means that our method is able to basically predict from the expectation value of the 2 upper microswitches what is the experimental value. And in fact, for the blue ligand here, BI167107, and for carazolol, we didn't have the experimental value, so we just used the value of the correlation that we found. And we were able to see that BI167107 is predicted to be a full agonist, whereas carazolol is predicted to be an antagonist, and this is exactly what they are characterized like experimentally. So that was really quite beautiful. And then finally, I want to finish with this aspect. So like I said already, on the plots along the microswitches, we could see that the active-like states are quite different from one another. And so we sought to see in which ways they were different. So here what we do is that we take the active-like state ensembles, and then again, we use dimensionality reduction and machine learning methods to learn in which way they're different. When we use dimensionality reduction, whether it's linear or nonlinear, we can see that the agonists tend to cluster together, but that the active states occupy a slightly different region of conformational space, so as expected. And we can then learn using these machine learning tools in which what were the distances or the important input features that are important for distinguishing active-like states. So for example, here we train the classifier to distinguish agonists versus non-agonists, and then we can see that the regions that pop up as important for this are the TM5 bulge and the NPXXY motif, as well as the TM6 moving outwards. So this is exactly what we expect. But what's quite interesting is that when we run the same classifier on all ligands, how do we distinguish these 7 conditions? We can see that the TM5 bulge here doesn't appear. And so this means there's too much conformational variability between the ligands to be able to notice this difference. You only see it when you try to cluster agonists, not cluster when you try to distinguish agonists versus non-agonists. So when we take these results together, we're able to come up with a model of activation that is quite unifying. And basically, we can see that the agonists exercise direct control on the TM5 bulge, and that gets propagated then to a connector region and interactions between TM5 and TM7, and that leads the NPXXY motif to take a unique conformation, as well as the TM6 displacement. In addition to this, all the ligands exert a direct control on TM7 on the allosteric control, but not agonist versus non-agonist. And I think I'll skip this last part that was really showing the molecular details. I think I have said quite a lot, but just know that we can go quite deeply and then look at very detailed parts of the system, which could be important for drug discovery. So I'll conclude with this. We have realized as we were categorizing these enhanced sampling techniques that they hinge only on a very few key principles that we organized in this decision tree I showed you. Most successful techniques seem to combine different ingredients in an advantageous way. It's important to have an overview of enhanced sampling techniques and of the, because in order to pick one, we need to basically know the system and compare it to what the different enhanced sampling techniques enable. So with an overview of the methods and of the principles that underlie them, then we can avoid pitfalls, hopefully. It would be very useful to systematically compare the methods. So we started trying to do this by unifying the notation and the language that we use, but we're still quite a long way away to being able to say systematically what method is more efficient for which one. And for this, I think we lack a set of realistic benchmark systems. It would be, of course, one of my dreams to be able to come up with a workflow for complex problems. And so with this, I would like to end. And I would like to acknowledge, on top of Jérôme Ménin, the other people who have been involved in the enhanced sampling sorting project. And then the GPCR activation project was done in my lab, mainly by Oliver Fleetwood, a previous PhD student, together with also Annie Sergio Yua. And this is a collaboration with a group of Jens Carlsen at Uppsala University. And of course, like all the others, I'm very grateful to the rest of my group, to funding sources, and to bodies that provide computational time. And thank you for your attention. And I don't know if I left enough time for questions.
We can definitely take one or two. While we are waiting for people to populate the chat, I have a question. So you showed this linear correlation between the CAM production and the distance. I'm some sort of collective variable, I guess. But I'm sure that there must be the experimental data that's available for CAM production would be the experimental value, I guess. But you would have calculated the free energy that could be correlated, I guess, right? Straight away.
Yeah, so we did, if I understand your question correctly, so on the y-axis is the experimental value, and then on the x-axis we use the expectation value. But we have also calculated the free energy difference between the two states. So expectation value is just one way to measure the balance between the different states, pretty much. And the reason why we report this instead of the free energy difference is that it's sometimes different to determine the bounds of the states. So for example, on this sort of landscape, do you consider this part of the activated state or not? that makes it quite difficult to just calculate the delta G between the two. So we find that by calculating the expectation value, we don't really have to decide what is part of a state.
Right, okay. So Biman asks, does the swarm of trajectory method will be able to capture multiple pathways if it exists?
Yeah, no, that's a really good question, remark. So in fact, you're right that the string method basically really captures one path, and it captures a path that is also decently close to the initial path. And so that's why for this specific method, I think, or for this specific problem, we were in a really good state because we had some initial trajectories. But imagine if we start from a path that is really way off and the actual path we're interested in is separated from that initial path by a free energy hill, then we would never find it. It's the optimization hinges on finding the drift away from the initial path.
Okay.
And just to mention, so I think the quantitative agreement with the experimental value shows that probably we have captured the main path. But what I would recommend in the most general case, if we don't know that the conformational change is dominated by one pathway, is either to not use the string method with swarms of trajectories, or to just repeat the calculation with different initial pathways to just make sure.
Okay. So Nishant asks, can you give some more details about how the optimization of the pathway is done?
Do you mean the method in general? I guess that maybe is the question.
That he's asking about the details about the string method, I think.
Yeah. So are you using the string method, or is it done? So is it just a direct minimization?
No, so the string of swarms is based on initiating many short trajectories from a single point, and calculating the drift, the average drift of all of the trajectories, and then placing basically the next iteration of the string, then the pathway is being displaced towards where the drift pointed towards. So that's how you end up in a minimum valley.
Okay. I see. Okay. All right. So you're not really computing forces and moving the beats, right? I mean, unlike.
No, this is more simple than this. So basically you just use the drift. But that's why it's really, the initial path really matters, because basically you're never going to be able to overcome barriers that way.
Exactly. Yeah. Okay.
Right. Okay. Yeah. Okay. So I think if there are any more questions, I invite the participants to put it in the chat, and Lucie will definitely answer them. I thank all the speakers for